Hi, good evening from Rome. I'm Michela Rossi, and thanks to invite me to present our data about the CLA grant that I've received last year. I will create a short picture of me to share with you my presentation, and at the end I will recreate a big picture. So let's start with the presentation. Here it is. So I can start now. My work is about the analysis of a new therapeutic approach to inhibit the osteolysis in Gorham Stout disease that, as all of you know, is a rare disorder described for the first time in 1838 by Jackson and characterized by angiomatous proliferation and progressive osteolysis without new bone formation. So far, only 300 patients are described in literature. Due to its rarity, the etiology is still unknown. The diagnosis is based on histological examination, and there are no set therapeutic approach to treat the disease. In our previous works, we investigated the bone remodeling activity in GSD. Bone remodeling is a physiological mechanism that aims to maintain healthy bone. It is based on different phases in which giant multinucleated cells, osteoclasts, resorb old and ischemic microfractured bone, and osteoblast later, the bone-forming cells, rebuild the new one. At the end of the bone formation phase, they can become lining cells covering the bone matrix, or they may be entrapped into the new bone mineralized matrix, and they differentiate into osteocytes. Bone remodeling activity is tightly regulated. Indeed, alteration of this mechanism lead to increase or reduce bone mass, as in GSD. For our studies, 10 patients were enrolled by the rare disease program of our hospital. They are from 1 to 31 years old and are in prevalence males. They display osteolysis both in the axial and appendicular skeleton, and 2 of them display also chylothorax, that is the most rare but severe manifestation of the disease. We firstly performed histological or histomorphometric analysis of bone biopsies of patients, revealing the presence of fibrosis in the bone marrow stroma and increased number and area of vessels, increased osteoclast surface and number, and increased osteocytic lacunae dimension. Moreover, we reported an alteration of some serum markers as bone resorption marker ICTP, sclerostin, interleukin-6, and VEGF-A that exert a stimulator and inhibitor effect on osteoclast and osteoblast respectively. To deeply study bone cells, we perform in vitro experiments. PBNC isolated from patients display an increased ability to differentiate into osteoclast with increased number of nuclei per cell, even more than 30. To dissect the mechanism underlying the increased osteoclastogenesis, we recently analyzed the circulating population of osteoclast precursors in GSD. And FACS analysis revealed that despite no alteration in the monocytic population, GSD patients present increased CD16 negative population that, as reported by Comano and co-author, is a population more prone to differentiate into osteoclast. Furthermore, in GSD, we revealed also an increase in the percentage of CD115, the receptor of monocytic colony stimulating factor that stimulates the proliferation of osteoclast precursors and induces the expression of rank receptors. Next, we evaluated the bone resorption activity. First of all, we observed that About the 75% of GSD osteoclasts showed a more motile phenotype compared to control, as revealed by Faludin staining, and that GSD osteoclasts resorb bone more than control cells. To understand the altered molecular mechanism involved in osteoclast dysfunction, we perform a gene expression analysis that revealed a modulation of 106 gene in GSD osteoclast. A deeper analysis revealed that the significantly modulated genes are involved in angiotensin II, PI3 kinase in AGF receptor signaling pathways. Interestingly, all these pathways are related to PTEN, that is involved in osteoclast differentiation and migration. Preliminary results confirm a reduction of PTEN expression in GSD osteoclast by real time and by immunohistochemical staining. To complete the molecular analysis on osteoclast, we performed also a myeloma analysis that revealed an increase of 3 microRNA, microRNA 1246, microRNA 1, and microRNA 137. All those 3 microRNAs are involved in PI3 kinase and MAP kinase pathways. Since a feature of GSD is the absence of bone formation after bone resorption, we evaluated for the first time the involvement of osteoblasts in this disease. And we had the opportunity to isolate mesenchymal stem cells from one patient, and we observed that these cells displayed the same spindle-shaped morphology of control and the same immunophenotype of control cells. We tested the ability of patient MSCs to differentiate into osteoblasts, and as you can see by the alkaline phosphatase staining, there's no alteration in the differentiation ability of GSD cells. However, GSD osteoblasts display a defect in the mineralization of nodule formation. Moreover, GSD osteoblasts had increased osteoclastogenic potential as revealed by the rank ligand and OPG ratio. Rank ligand is the main osteoclastogenic cytokine and osteoprotegerin is its decoy receptors. As for osteoclast, we perform a large array analysis, although we have cells only from one patient, and we identified 72 modulated genes that are involved in extracellular matrix organization, bone morphogenesis, and interestingly, the most upregulated gene was MMP13, confirmed also by real-time expression analysis. MMP13 is a matrix metalloproteinase that is involved in several pathways in angiogenesis and osteocytic osteolysis that could explain the increase of osteocytic cytic lacunae that we observed in bone biopsies from patients. Also for osteoblast, we performed the molecular investigation with the myeloma analysis, and we identify 4 microRNA, but these data are really preliminary because we have cells only from one of our 10 patients recruited for the studies. The regulation of bone remodeling is not only organized by bone cells, but is a complex mechanism involving also systemic factors such as growth factors, hormone, extracellular vesicles, and the interaction with cells of other compartments such as immune cells. In 2021, briefly, we demonstrated in GSD patient a reduction of regulatory T cells able to inhibit osteoclastogenesis. This is just to inform you on how the bone remodeling activity is a very complex mechanism, and recently many papers by our group and others suggest that it's also regulated by extracellular vesicles. Therefore, we characterized also in GSD this heterogeneous group of small particles, bilayered particles released by all kinds of cells made of exosome and microvesicles that are different for dimension and biogenesis. These particles may vehicle nucleic acid, protein, even organelles, and could be found in the microenvironment or in the circulation and can influence and interact with target cells in 3 different ways: interacting with the receptors, fusing with the plasma membrane, and by endocytosis or internalization. We isolated extracellular vesicles from plasma of GSD and controls, and we analyzed them by electronic microscopy. The analysis revealed that they are rounded shape and that they could be present in cluster or single particles, and they have a dimension range of 100 nanometers, suggesting an enrichment in exosomes. We then performed FACS analysis of extracellular vesicles isolated from plasma of GSD patient and sex and age match controls. After staining with intracytoplasmic green dye that let us identify the population of interest, we quantify the number of extracellular vesicles in each samples, and as you can see, there's no difference between patients and control. Since previous results obtained revealed increase of rank ligand expression and increased osteoclastogenic potential of GSD cells, we investigated by FACS the expression of rank ligand in the membrane of these vesicles. And interestingly, as you can see by the graph, GSD patients have more rank ligand positive circulating extracellular vesicles compared to control. Proteomic analysis of those vesicles let us identify 94 proteins that are expressed in GSD extracellular vesicles. These proteins are again related to PI3 kinase pathways and AGF receptor angiogenesis and T cell activation. Furthermore, microRNA 1246 was the most upregulated in GSD extracellular vesicles. Looking at our previous works, we decided to test a new approach to inhibit osteoclast function and reduce the osteolysis in GSD, modulating one of the pathways that I previously cited. I'm so sorry that I cannot be more precise in the description, but we have a pending patent application and we have to keep the secret about it. So I will use from now on the general term molecule to identify the target we are interested in, and that is modulated in GSD patients, and the general term drug to identify the chemical that we are testing and that is under patent application. We first of all demonstrated that the molecule we are interested in, never investigated in bone before, was increased during osteoclast differentiation, suggesting a role in osteoclast function. Indeed, if we treat LT donor PBNC with a chemical that stimulates or reduces the production of this molecule, we obtain an increase or a reduction of osteoclast formation, respectively, confirming an important role of this molecule in osteoclast. Due to the increase of this molecule in GSD patients, we induce the differentiation of PBNC isolated from a patient with the drug that we are testing, and with a reduction of the molecule expression, we obtain a reduction of differentiation and activity marker of osteoclast, such as TRAP, DC stamp, and CLCN7. Moreover, the drug reduced the osteoclast formation of GSD PBNC to levels similar of control cells, as you can see also by the TRAP staining. My CLA grant is still under approval, but further experiment will be focused on two main aims. First of all, we will characterize the role of the molecule in the bone physiology, and then we will use the drug on GSD osteoclast to inhibit our molecule of interest expression to investigate its potential as new therapeutic approach for GSD patients. So I would like to thank my mentor, Dr. Del Fattore, the head of the bone physiopathology unit of Bambino Gesù Children's Hospital, and all my colleagues, our collaborators, our agency that let us work, and all of you for your kind attention. Thanks, and thanks again for the invitation, and I hope to be there with you in the next years with a new grant and with a new proposal to discuss finally in person with you. Thanks. Bye.